everyone. Welcome to the webinar titled Proven Strategies to Boost Your Protein Expression. Today we will discuss how to maximize yields without compromising quality. I'm Hélène Meyer from Proteogenics and I will be your host today. We are very fortunate to be joined by Raphael Hoffner, CSO here at Proteogenics, and Clément Aubin, our Global Product Manager. We encourage you to ask your questions during the webinar using the chat. At the end of the presentation, we'll have a dedicated Q&A session where we will answer as many questions as possible. So to start, before we dive into the topic, let's start with a quick warm-up. So I have a few questions for you to ask today, and I would love to see your answers. So don't hesitate to participate. So the first question is, for what uses do you need to produce proteins? And this is a multiple choice type, so you can have several answers. Well, we have many participants today. This is great. The second question is, what challenges do you face when producing proteins? And the last question is, what system do you use? And it is also a multiple choice question. Well, we have a lot of answers. Thank you so much. Okay, thank you for participating. So now that this is done, I'm happy to welcome Clément Aubin, our Global Product Manager. Clément, thank you so much for joining us today. And the floor is yours whenever you are ready.
Thank you, Hélène, for this nice presentation. Welcome, everyone, and thank you for joining our webinar today. I'm very happy to present you this webinar on proven strategies to boost your protein expression, combining high yields and optimal functionality. So today we'll focus on transient gene expression. We will first explore why transient protein expression is important in today's research. We will then get deeper in the understanding the key challenges we face in transient gene expression, from the choice of the right system to the optimization of the culture system in order to maximize the yield while optimizing the functionality of the obtained protein. We will also explore scalability issues. And finally, we will introduce our exceptional brand new system, extension-race, and show you how its performance can tremendously improve your research purposes. But before we do this, I'd like to introduce you to Proteogenics, which is a leading CRO in biologics discovery and development. So it was founded by Philip van Vrock and Dr. Raphael Hoffner, that you will have the pleasure to hear in a few minutes, in 2003. And it was established in Strasbourg, or near Strasbourg in France. And today we've grown to 160 employees and have over 22 years of expertise in advancing biologics research. So our services cover full spectrum of biologics discovery, from gene synthesis and peptide synthesis to polyclonal and monoclonal antibody development. And also we have a full catalog of products, mainly proteins and antibodies. So Proteogenics is your trusted partner to deliver scalable and high quality solutions, so from gene to product, and to support your research and clinical goals. So now that you know a little bit more about Proteogenics, let's explore shortly our comprehensive pipeline of antibody services. So it offers an end-to-end solution from discovery to bioproduction. And this seamless process ensures our customers don't have to coordinate multiple vendors to bring their therapeutic antibodies to life. So It is the path. And antigen design and production is the first step for this. So we provide peptide synthesis, gene synthesis, and protein expression. Then for antibody generation, we offer polyclonal and monoclonal antibody through hybridoma and single B cells and phage display technology. Then after generation, antibody analysis follows, ensuring quality through epitope mapping, affinity and stability evaluation, and of course purity assessment as well. For sequencing needs, we offer hybridoma sequencing and mass spectrometry. For the antibody engineering stage, we provide affinity maturation, antibody humanization, and AI-based, artificial intelligence-based developability improvement, and the creation of bispecific antibodies, as well as antibody drug conjugates. Finally, during bioproduction, we ensure efficient high-yield production with stable cell line development, and we also on the other side of the line provide high throughput production, and as well transient mammalian expression, which is our topic today. So by partnering with Proteogenics, you ensure speed, quality, and consistency throughout the entire antibody development process, saving time and reducing the risk of errors and delay, and also the management of different vendors. So to begin with our topic today, so the path to developing a therapeutic antibody is complex and involves several critical stages. First, the process begins with the discovery phase, including various antibodies generation, their screening and selection, and the chosen lead optimization. In preclinical phase, the focus is made on in vitro and in vivo studies to confirm the properties of the lead previously chosen, and the process development to come up with a robust system enabling a perfectly functional antibody. In early stage production phase, scale-up is validated to check that all characteristics previously optimized are kept while producing high quantities of antibodies. In the same manner, good manufacturing practices like production is set up and investigational new drug studies are conducted prior to going into clinical phase. And what we see here is that in most of these critical steps, transient expression is required. Indeed, as multiple productions are required following incremental optimizations, transient expression offers the best solution to generate the antibodies, screen and select, optimize them, and produce them again, as well as to conduct the in vitro studies, and it may also be useful in scale-up processes to optimize it as well. So why is that? Among the strengths of such production method, we have to note that its speed and flexibility makes it easier to develop the antibody, change and adapt it, and produce it again. So multiple testing in parallel is also enabled, which is key in screening and selection to save time. Since the development of transient expression on the other side, since this development does not need extensive engineering, the costs implied before the very beginning of antibody generation are lower, and you may begin your first antibody production faster. Finally, transient expression is very scalable as it adapts with a high productivity each time from small R&D projects to large-scale productions, from microgram to near gram scale. So to set up a transient expression system, some key aspects need to be taken into consideration. The first one is the choice of the cell line to be used. So bacteria, such as E. coli, offer affordable and fast production options. They are ideal for producing simple proteins with high yields and in fast productions. However, it lacks the post-translational modifications that may be needed for more complex therapeutics, such as glycosylation, and they are also harder to purify. In the case of yeast expressions, it is also quite cost-effective and offers an unmatched scalability, but it has low glycosylation as well, and it also shows higher risk of plasmid loss. In insect cells, on the other hand, offers flexibility for producing certain proteins, particularly those that require complex post-translational modifications. However, they may not always be the first choice, especially in therapeutics development, due to difference in glycosylation with human proteins. They are therefore especially used and preferred in animal health studies. And finally, mammalian cells are the preferred system for the very high yields, their adaptability to most proteins, and their human-like post-translational modification. They do carry some cons, such as their higher costs, their need for CO2 management during culture, and the risk of human viral or mycoplasma infections for some of them. But the advantages of the high yields and human-like post-translational modifications do compensate these technical constraints. So for the rest of this presentation, we will focus on this type of cells. So among the mammalian cells, Chinese hamster ovary cells, so CHO cells, are often the preferred ones. So they are one of the most widely used cell line in biologics productions, especially for complex proteins and antibodies. They have a well-established history for producing high-quality, scalable proteins, and about 80% of approved human therapeutic antibodies are produced using their cells. So CHO benefits really from a richer literature and references of validated antibodies. So if we now compare them with human embryonic kidney cells, so HEK cells, the second mammalian cells in order of use, CHO offer a better and easier scalability, as well as a better transition from transient to stable expressions. So this is especially useful when moving into production phase. So in the end of the therapeutic antibody development process. CHO also show a lower risk of human virus infections since they are non-human cells, and so biosafety requirements are lower. So, and CHO cells are also preferred ones, so globally, for those three main reasons: their high yields, their scalability from small batches in a plate to large bioreactors, and their adaptability to bioproduction type. Before we get deeper into the technique, let's recap the main steps of transient expression. In the first place, cells are selected, as seen previously, and prepared for culture. The other upfront step is the plasmid design. So it's the preparation of the vector and sequence that will be expressed. The transfection gives the start of culture and leads to careful monitoring of proteins that are expressed. Once the culture time is over, proteins are collected and purified before the careful analysis. Parameters and protocols of each of these steps are crucial to get the maximum of proteins in the right format. And I now give the stage to Dr. Raphael Hoffner, who will show you how extension rays can help you go through this process.
Hello, everyone, and thank you, Clément. Thank you also, Hélène, for the kind introduction. Before to start, I just wanted to say that I am happy and proud to have the opportunity today to share some information about this new extension array cell line. It was developed by our R&D team based here in France, in Strasbourg, and the creation of this cell line has been a long and challenging process. Today, I will not go into technical details of the creation of the cell line itself, but I will share some key principles that guided the development of the extension arrays expression system as a whole. Of course, I will also show some protein production data. So, extension array cells were developed using a CHO-K1 cell line as a foundation. CHO-K1 is widely used for the production of biopharmaceuticals with stable cell lines, particularly recombinant protein like, for example, antibodies. As you may already know, and as Clément already mentioned previously, CHO cells have advantages for stable production as detailed on this slide. I will just mention again their robust growth in suspension cultures, simplifying large-scale production in bioreactors. Their genetic stability making them ideal for consistent recombinant protein production, and their resistance to viral contamination compared to human or other mammalian cell lines. CHO cells have a lower risk of viral contamination, improving the safety of biologics. It is also important to mention that they can produce glycosylated proteins with patterns similar to those found in humans, which is crucial for stability, efficacy, and safety of biologic drugs. Last but not least, they can also offer high production yields in stable cell lines. However, one major weakness of CHO-K1 is their relatively poor performance in transient expression, especially when compared to HEK cells, which is more commonly used for this application. This limitation hinders the use of CHO in the early stages of therapeutic molecule development. Ideally, using CHO from the start would prevent unexpected issues related to final product specifications, which can differ significantly between HEK and CHO in terms of activity and post-translation modifications. Having access to a CHO cell line with high transient expression performance would solve this issue. This is precisely why Proteogenics decided to work on CHO-K1 engineering in order to develop the extension array system. The extension array cells are designed to outperform existing systems in terms of yield, while also ensuring that recombinant protein quality meets the requirements of therapeutic molecules in terms of purity, activity, and PTMs. The need for a CHO cell line with enhanced transient expression capabilities is clear. But why did Proteogenics decide to tackle the challenge of creating such a cell line? And how did we proceed to succeed? The answer of the question why is simple. We have over 15 years of expertise in cell line development and engineering. We were confident that we could make significant improvements to CHO-K1 capabilities for transient expression. However, the journey to answering the question how was more difficult. It took us a total of 8 years of R&D to finalize the extension array system, including 3 years of internal validation before releasing it to our clients. With extension array cells, we have successfully achieved two main objectives. We have modified and optimized the CHO-K1 cells and have succeeded to overcome low production yield in transient expression, and we have reduced the production time. In 2022, we were first able to offer to our client the extension cell line, which already outperformed CHO-K1 in terms of production yields. And now in 2025, we are proud to introduce its successor, extension arrays, which enables even higher production yields and reduced production times. As mentioned in the introduction, I will not go into technical details in the creation of the extension array cell line. I will outline some key principles that guided our approach to the development of the extension array expression system. Extension array cells are, of course, the core of the expression system, but our expression kit is not only optimized at the cell line level. Achieving optimal and maximum production yields also requires the optimization of the transfection. If this aspect is not optimized, you will have a poor plasmid DNA penetration into cells or an early plasmid loss, leading both to low expression and also highly variable yields. Moreover, obtaining maximal production yields also depends on optimizing the expression cassette within the plasmid DNA. If the promoter and other untranslated regions are not optimal, or if the cDNA sequence is not optimized, the transcription level will be low, resulting in low recombinant protein expression. At Proteogenics, we have addressed these challenges from the very start of the project by designing the PXN1 vector as an ideal solution to overcome all these issues. This plasmid works in synergy with the optimization of the CHO-race cell line, and using it guarantees maximum performance of our expression kit. Here is the first example of what we obtain when producing a therapeutic antibody, a human IgG1. As you can see, after only 8 days of production, we achieve a yield of 250 meg per liter after protein A purification. But the production duration can also be extended to 15 days to exceed 300 meg per liter. This means that with our expression system, you have the flexibility to choose either a short production time with high yield or to extend the production duration to maximize the yields. To maximize recombinant protein production, beyond the expression plasmid itself, it is also essential to optimize the transfection protocol. This includes the handling of the cells in optimal conditions both before and after transfection. A second key factor is to avoid suboptimal culture conditions. It is crucial to optimize pre- and post-transfection culture conditions and, of course, use a high-quality culture medium that is optimized for protein production. At Proteogenics, we optimize every step from cell sourcing to pre-transfection culture, transfection itself, and post-transfection culture. We have also carefully selected and validated the optimal culture medium to achieve the highest possible performance. Here is a second example of what we observe for the production of another therapeutic antibody, a human IgG4 garetosmab. This example is in fact currently the highest production yield that we obtain with extension array cells. I want to emphasize that this was achieved using a simple flask production system and that the results can be consistently replicated since we obtained the same results in 2 different production batches. As you can see, with extension arrays, it is possible to reach a production level as high as 1.8 gram per liter using this straightforward flask culture setup. As previously mentioned, we conducted 3 years of validation before launching the extension array system. These validations covered multiple key aspects, and one of the first being the ability to scale up production efficiently. In fact, we have been using extension arrays for over 2 years in our internal protein production projects, covering a wide range of protein types and species. Through this experience, we have validated the consistency of yields and product quality at different production scales. These different scales can match with various stages in the development of a therapeutic molecule. Mini-scale expression in plates can be used, for example, for affinity maturation candidate screening. Medium-scale expression in flask is suitable for in-depth candidates studies. Small to large-scale expression in flasks or in bioreactors up to 10 liters or even 20 liters in our labs can be used for preclinical and early clinical development. Beyond yield consistency, our 3 years of validation efforts also focused on assessing and validating product quality obtained with the extension array system. To conclude my presentation, I will now illustrate this in a few case studies. This first case study illustrates a comparative glycosylation profile analysis of the therapeutic antibody zolbetuximab, a humanized IgG1. 2 samples, one produced using stable SHOK1 and the other one produced using extension arrays, were analyzed by spectroscopy. The results obtained show on the left side very similar glycan profiles between the 2 samples, and on the right side, highly similar sugar content. This confirms that extension arrays allows for rapid and high-yield recombinant protein production without making critical compromises on glycosylation profile. This second case study focuses on quality controls for integrity, purity, and aggregation using PAGE and seq-HPLC analysis. Here, a recombinant human IgG1 was produced for an R&D project. Despite a high production yield of almost 300 meg per liter, the integrity and purity of the final sample obtained after a one-step protein A purification is remarkable, over 99%. And we obtain an extremely low aggregation rate observed on the HPLC profile. This third case study illustrates very similar quality controls for integrity, purity, and aggregation using again PAGE and seq-HPLC analysis, but this time for the production of a mouse IgG1. Again, despite an impressive yield of 250 meg per liter, the purified sample exhibits exceptional purity, close to perfection, and with a very low aggregation rate as shown on the HPLC profile. These 2 case studies I just showed you highlight how extension arrays delivers high-yield antibody production without compromising protein integrity, purity, and aggregation, making it an excellent choice for producing both human and mouse antibodies, which are widely used in the biotech and pharma industries. I will conclude my presentation with this 4th case study. It presents a comparative ELISA activity analysis of 3 different antibody samples. The reference antibody in this study is the 72D9 produced using an hybridoma developed by Proteogenics, which we have subsequently sequenced. The antibody was then produced using extension array cells with the same sequence as the original hybridoma, named antibody 1 on the graph, as well as with a slightly modified sequence named antibody 2 on the graph. As you can see from the dose response curves, the recombinant antibody produced in extension arrays is just as active as the original hybridoma produced antibody. In fact, if you looked at the EC50 values calculated from the ELISA data, it even suggests a slightly better activity for the recombinant antibody produced with extension array cells. This final case study demonstrates that extension arrays can efficiently produce recombinant proteins with great activity levels, and in this example, identical to that of the original molecule produced by an hybridoma. I thank you for your attention, and now back to Clément for the next part of the presentation.
Thank you, Raphael, for this precious insights and case studies. So we have seen that with garetosmab, extension rates could reach the top record of 1.8 gram per liter. It is not a single success of extension race, for it outperforms the market leader that you surely know for all the tested antibodies. So it is even more than 6 times better in some cases in terms of yield, such as with 602-mumab, which is here in the middle of the screen. So main antibody isotypes have been covered from IgG1 to IgG4 and IgA, for example, giving similar results of great productions. So extension race has been designed for this purpose, top yields in transient expressions. Another aspect that was tested is as explained by Raphael, is the scalability of extension race cells. Here we can see that both in 96 well plates or in flask, extension race always deliver higher yields. We see that it is especially well performing in plates, which is ideal for screening. So you do not have to use a full flask and associated material and reagents to get enough proteins for your analysis anymore. Antibody screening and selection is easier and more cost efficient also. So, and all in all, if we compare extension race with market leaders' solution, we have yields that were about twice higher on average. And with if we compare the cost of the extension race kit with its competitor solution, plus the fact that with less material you have more produced proteins, well, the cost per milligram of protein expressed is about 3 times lower. So we calculated a cost of 3.8 euro per milligram of produced protein on average. And that's not it. For extension race comes with a short post-transfection culture that helps you save time. So you can harvest the protein in the same week as the transfection. In plate, the early harvest can be done as early as on day 6, and it can be on day 8 for flasks. So indeed, yields are higher when you use the extended plasmid retention, as you can see here. But even when you use the short protocol, yields are higher than with the competitor solutions with their long protocol. So you see here 237 compared to 134 to 35. So now you're now free to choose to save time while still expressing high amounts of antibodies. So you may have understood that this precious tool, that is extension race, helps our services and R&D department for some time now. It is now available for you. We offer extension race in the format of a kit containing all the main component that you need. So the cells, of course, but also the expression vector, 1 liter of medium, as well as transfection reagents, so that you do not have to source components that were optimized, and these components were optimized to work together to provide the highest yield. So you don't have to source from different vendors and create your own system by yourself. And you have enough material in the starter kit so to cultivate 1 liter of culture. The kit also contains a positive control so that you can test your culture conditions. And every component is also available separately, of course, for reorder. To have a little focus on the expression medium, please note that it is serum-free, animal component-free, and chemically defined, so that you have safer handling, lower contamination risk, which is very critical for therapeutic antibody development. And it is also in line with our commitment to reducing animal use in ethical and sustainable development. So this extension race can be within your hands in a few days, since it is already available and it is in stocks. We deliver worldwide in less than a week. So you can also benefit from a discount until end of May with promo code XTEM20. And indeed, the kit comes with full protocol and guidance. The user guide is also provided online, and we will send you the link to the page. And our support team is available to help you and will be very happy to answer your questions. Please do not hesitate to contact us, and I thank you very much for your attention. And we are ready to answer to your questions.
Well, thank you so much, Clément and Raphaël, for the great presentations. So with that, we move right to the Q&A session. So as a reminder, if you have a question you would like to ask, you can submit it in the chat. So let's jump now with the first question. So maybe it's for Raphaël. What precautions need to be taken while upscaling from high throughput scale to small scale?
Well, I'm not sure to fully understand the question, but if the question is it difficult to upscale from plates to flasks? The answer is no. In the datasheet that we provide with the kit, you have a full description of ideal conditions to be used either in plate or in flask. And we give advices to adapt DNA quantities and culture volumes and transfection protocols for each of culture formats.
Okay, thanks. So maybe the person who has the question can just say in the chat if it's the answer he was looking for. So another question for you, Raphaël. Have you tried other vectors with this cell line, and did you compare it with your vector? Is it essential to use your vector?
Well, is it essential to use PXTEN1 vector to have the maximum performance? That's for sure. That being said, using any mammalian cell expression vector with a PCMV promoter will be fine to have a good production yield. The only thing is that you will not have an extended culture duration that will be available. So you will rather produce within 8 days only, and you will probably have more plasmid loss.
Okay, well, thanks. Thanks for the answer. Here's another one for you. Is this kit optimized just for antibodies? May we use it to express recombinant proteins other than antibodies?
Yeah, of course, you can express other proteins. We have already tried during the past years many different types of antibody and antibody derivatives. So VHH, Fab, FC fusions, all kinds of nanobodies, VHH, SCFV, many different formats of P-specific antibody, and also other type of proteins like HLA proteins, CD proteins, mannose binding proteins, VEGF, neurexin, decorin, come into my mind, GP proteins, COVID-related proteins, MIC proteins, different types of enzymes that exhibit a good enzymatic activity also. So yeah, you can express antibodies with very high yields. Of course, other proteins with less high yields, but this will mainly depend on the molecule that is considered and the design of the molecule and of the expression strategy. Because of course, for mammalian cells, it's better to use a secreted expression strategy to avoid intracellular toxicity.
Okay, and maybe I can combine with this question. Is there a big difference in yields between antigens, antibodies, and Fabs?
Difficult to answer directly to this question because the yield can vary very much even between antibodies. It always depends on the optimization of the molecule. But for sure, Fab will be expressed at a lower level than classical IgG, full IgGs.
Okay, so there is another question about the yield. Is there a big difference in antibody yield between extensio, I think, and extensio race between the previous version and this?
About 20% more yield within a shorter production time.
Okay, well, thanks. So maybe this is a question for Clément. How much does the license for the kits cost in case we want to produce antibodies to our own customers?
Well, this is thank you for this question. It is indeed an important one. So, as you have understood, the kit comes with the right limited license, the right to use it for research use only, and then we can discuss indeed for a license for if you want to sell the produced antibodies. So this is discussed each time depending on the project and what the project is. So it depends. It is usually created based on the first signature fee and then royalties. But this is discussed depending on the project of each customer.
Okay, so maybe there's another question about the yield here. Yes, extension race media. No, it's not about the yield. Extension race media can be used for both CHO S cells and CHO K1.
Yes, it can be used. Yes, it can be used for both cells.
Okay. And does your medium post-transfections contain drugs that inhibit cell divisions, mitosis?
That is a question that we cannot answer.
Okay, so maybe it's a good opportunity to get in touch. So Lisa asks, if you buy the kit, is there also helps provided by you for optimization of the plasmid and antibody design to have a high production as possible? Maybe Clément, you want to answer this?
So I think that it was if you use your own vector, I guess the question is. So if it is so, well, Raphael answered that indeed if you want the highest yield possible, then the better solution is to subclone your gene into our vector, into a PXTEN1 vector. But in case, but we offer also this service, so we can do this for you as well.
We can also propose to optimize the cDNA sequence. We have recently developed internally an AI-powered codon optimization solution that can help to optimize the production as much as possible.
Okay, well, thank you. Maybe for you, Raphael, we have a question here. Are the antibodies produced with extension race functional for ELISA, Western blot, others?
Yeah, of course, we have, in most cases, yes, we have expressed functional antibodies in all types of applications, including ELISA, Western blot, flow cytometry, immunocytochemistry, all technologies for affinity measurements, BIACOR, SPR, and BLI. And we have also produced, as I said previously, a number of enzymes, such as myosim and sulfatases, come into my mind, which have been shown to have enzymatic activity equivalent to commercial reference products.
Okay, thank you. And Clément, can you explain how to smooth the change in expression system?
Well, yes. So indeed, it was. We often receive that request from customers. So because it is not always easy, so to change your expression system. So we offer different solutions. So of course, our support team is always available. We have highly simplified our protocols, and we have also launched tutorial videos, so that can be delivered also day by day after your transfection day once you warn us, and so that you can easily follow the steps of the protocol step by step without always coming back to the protocol and studying it deeply. And of course, so the kit is delivered with a protocol that is waterproof, so that you can take it to the lab easily.
Okay, great. So Clément again, how to store and how do you deliver the cells?
So the cells must be stored in liquid nitrogen, and all the kit is delivered. So the bottle of medium is delivered into blue ice pack, while the other items, so the transfection reagents, the cells, are delivered on dry ice at -20 degrees, but they should shortly after that be stored into liquid nitrogens. And all the details of each item storage is given into the user guide.
Okay, there is another one here. Is there specific equipment that we should use? Maybe Raphael, you can answer this one.
No, you don't need any specific equipment. Any CO2 incubators can be used for the use of the cell line, assuming that you have an agitation system.
Okay, well, thanks. Maybe we'll end with more general questions. Question for Clément: what services are offered for lead generation for in vivo studies? Do you also offer transgenic mouse model studies?
So thank you for this question. So indeed, we're not maybe the best here to answer to your question. So you will have certainly to contact us so that you can redirect your question to the right colleague. But for the lead generation, so we offer services such as hybridoma with, for example, humanized mice, single B cell for highly specific antibody generation, and phage display, which is very fast and especially good for difficult targets. But once again, so if you need, and we invite you to contact us directly, so that we can explore what is your project and how we can help in depth, so that you have your right answer. And it's the same for the in vivo studies that my colleagues will be able to answer to you in a better way.
Sure. Okay, I think we'll end on this high note. But Clément and Raphael, thank you so much for your great presentations and all your insights in this Q&A session. It has been a pleasure talking to you today. So before you leave, we would really appreciate if you could take a few seconds to answer a short feedback survey. This is a 2-question survey. It's very valuable for us to help us improve for future webinars. So here is it. Thank you for your time and participation. We hope you enjoy the webinar, and we will see you again next time. Have a great day, great evening, everyone.
Bye-bye.
Thank you very much. Bye.